All right, welcome everyone to the to week 2 of our Foundations of Clustering Methods in Cancer Research. Really excited to talk to you about this. Last week we went over PCA and dimension reduction to kind of prepare ourselves for this next step, right? To get the data focused and pre-processed. So here we go. So clustering. What's the point of clustering? Like, it seems obvious to put things in different categories, but why does this matter in cancer research? Well, I mean, obviously, we've learned over the last, with single cell developments in the last, I'm trying to think how long I've been in research, the last 8 years or so, we really found out how heterogeneous cancer is, right? People used to be like, hey, we're going to cure cancer. What does that even mean? You've cured one subset of one cancer for one patient, right? Because cancer is not a single disease, right? It's hundreds of diseases. And even within one subtype, with one cancer like breast cancer, you have multiple subtypes and a lot of heterogeneity that will respond different to treatments. So how did we discover these in the first place? Discovering these 5 molecular subtypes for breast cancer has had a real clinical impact. So how did this happen in the first place? Well, just to give you kind of a starting point of how you can work on this kind of data, we will be able to work on real cancer data sets today from TCGA. I think that'll be really cool. And basically today our learning objectives are basically we're going to introduce the difference between supervised and unsupervised learning. We're going to apply k-means clustering and hierarchical clustering to a TCGA data set that we're going to normalize and everything by hand. And then we're going to evaluate the cluster quality between these different results using some internal and external methods. So let's get started setting up our cancer data set and libraries. So for our libraries, a couple new ones at PCA tools that we used last time helps out. DSeq2 for normalization, TCGA biolinks, really important package that we're going to go over today that has to do with downloading our data sets and summarize experiment. Also a way to, once you download some of these data sets, to organize them. And then other than that, a lot of the similar things we've been working with, we'll just kind of pick them up as we go here. So first thing we're going to do is download our cancer data set. So off the top, really important thing, when you're doing any kind of analysis like this nowadays, single cell expression analysis, differential gene expression analysis, clustering. When you want to work with cancer data, you really want the raw values, and then you can work on doing your own normalizations that fit in your specific packages. So in the old days at TCGA, they would always just give us the raw data. It was really easy. But now it's like really hard. It's like paywalled or behind all these walls. It's very hard to get. The only data you can get is the TPM data or the FKPM data, which is normalized by things like, sequencing, transcript link, things like that. And that's great, but like a lot of the algorithms you need to use, they require the raw data. So while you can use things like TPM and you will get a result, and it might be okay, it's not the proper way to work with these things. So you really want to be able to have a way to download your TCGA data raw. And normally you always have raw data, right? When you work in your lab and your samples, you have raw data. So you don't like TPM, you start working on them. You don't just half normalize them. So I don't know why they do that. It's ridiculous, to be honest with you. It's ridiculous. So let's see how we can download a TCGA data set, our raw counts. So for all this, I use the TCGA biolinks package. And the first thing you do is we're going to download from the GDC. And basically you do a query. You put your project, it could be a BCRA, COAD, for example, any TCGA project. And then what data category do you want? Do you want mutation data? Do you want copy number data? We want transcriptome profiling, expression data, right? And, there's our category, and this is our data type. And you can read more about that in the TCGA biolinks page of how to properly nail these, but they aren't very hard to do, and they pretty much are the same among different cancer types. And then once we have our query, we're just trying to look for all our available samples in that data set. Now, we could pull them all down. That would be fine. I just want to do a subset. So I'm basically pulling out of the available cases. I am just taking 30 from the primary tumor and 10 from the solid tumor for today's work, right? So now I've chosen those, and I'm basically just grabbing the barcodes of these, basically, right? And then I do one more, a final query here, because I'm changing my data a little bit, and I'm saying I want GDC query. I want you want to have my project name, data category type, just like above, but now I want a workflow type. If you don't add this line, you'll just get whatever default value they have, and you may not even know what it is. So you want to have a workflow type. What kind of counts, right? And you can get the TPM counts here too, but we don't want those. So we want star counts. Those are the raw gene counts. And then what barcodes do we want? Well, we want the barcodes for these cells. This is pulling out the barcodes for us. And then we finally do a GDC download on that query through their API, and we download our actual data. And once we have our data, we just run this assay command. That gives us our expression matrix, and we can do column data on the data to get our sample info and kind of use that as sample info just to basically organize our metadata, right? We want our sample labels that show, our actual either the order or the sample names and their associated designation of their tumor normal. We look at our data. We have our 40 samples, 60,000 genes because we have pseudogenes, things like that. And then we have our sample types here, 30 tumor, 10 normal. All right, so now we have our actual data downloaded from GDC. It's looking good. But as I mentioned, it's raw, right? What are we going to do with this raw data? How can we pre-process this? We can't just throw it in. So the first thing you need to do when you have this raw data is normalize for your library size and stabilize the variance, right? Well, luckily, DSeq2, which is a differential gene expression, very popular algorithm for that, does this in its own package where it normalizes for library size and stabilizes the variance simultaneously. So it's a really good thing for us. So what we're going to do is we need to make a metadata column. So here's the function basically we're looking at here. DSeq dataset from matrix, because we're pulling in a matrix, our own dataset basically, right? So we have DSeq dataset from matrix, our count data, which is our expression data, no big deal, rows, columns. Then we have a column data where we have to have our sample metadata. And basically this is just a data frame where the row names are the patient names and the columns are different attributes that you would like to normally try to tell the difference between things. Like let's say you had a column called type and it was normal tumor. You could do differential gene expression analysis between those two types. How we don't have that. We have no design here. All we care about is normalization. So we can just put design equals ~1, right? And if you want to see what that matrix looked like, I can look at that column data. I think I have it somewhere, right? Thought I would have that column data like ready to go, right? It's just at the top. It's the sample metadata, right? Sample metadata, just to show you what this looks like. You see here we need a column of row names, and we need row names, sample names as row names, and we need something here. So I just added cancer type and put TGA breast. Normally this would be where your parameters for separation would be and differential gene expression. But we just need to have something in this column. So this is just a something column. I basically just listed what we have, and we have our row names, and these row names need to match the columns basically on that expression data that we you see how the expression data, the columns are the same as the row names. So you need that to be kind of aligned to get it in this thing. So that's all I really did is I basically just took the column names as my sample names, and then I looked in, made a cancer type column. So I have basically my column names as my expression, as my row names, and then I added that cancer type column. That's it. Then I entered it into the algorithm here for DSeq2, and I got my DSeq2 object. And then what I want to do here, the whole point is to apply this VST transformation. So I just put the DDS in here, VST, bam, I have VST. Once again, I call the assay off the VST, and I finally have my actual expression matrix, okay? So this is my normalized expression matrix. Now, the next thing I want to do is we have lots of genes in here that don't have much of a signal. So let's find our most 500 most highly variable genes. And these are genes, if you looked across the expression level for one gene in every sample, how much do they vary across all the samples? Because the more variance, the more we'll be able to separate out different clusters and information, right? So I basically, just like last week, I applied a variance. I got the variance for all the genes, basically, in the expression matrix, and then I took my top genes as I basically sorted the variance, and where I had the top 500 with the most variance, right? So that gives me my top 500 most variable genes, and then I basically just subset out my expression matrix. I transpose it for later input, and then I scale it here. And by just putting scale with no arguments, I actually centered and scaled. All right, so I have my data downloaded from TCGA. I've normalized it, and I took the 500 most variable genes, and then I scaled it and centered it, okay? So now we have our actual gene matrix, expression matrix ready to go. All right, so that being said, let's jump into our first topic here, supervised versus unsupervised learning. What does that actually mean? The title kind of guesses it, right? But think of it this way. A supervised learning, you're a teacher with an answer key. For your data sets, you know the answers. Therefore, you have a label for every single cell, where it could be a cell type, tumor type, whatever. You have a label. That's the point. Unsupervised learning, it's like you're an explorer. I want to know the Grand Canyon. I don't have a map. I don't know where I'm going. I'm going to look at what's around me and try to guess what to do. It's going to be a data-driven a lot, right? So how can this apply to our cancer data, right? So let's first look at supervised learning. And we'll use a very, very simple example, doing a KNN classification model. So I think this is cool because we will be using KNN graphs next week for things like Leighton clustering. And you also use KNN graphs for the UMAP, things like that, and TCD. So these network graphs are good to form a foundational interconnection matrix for other algorithms. Anyway, so I'm going to do a very, 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 very simple, almost trivial, well, not almost, it is trivial, implementation of KNN, just to kind of see what we mean when we say we have supervised learning labels, right? So we know the labels. We know they're tumor versus normal. So can we train a model now to guess that on unseen data? Okay. What we're going to do is we're going to split our data set into 70%, 30%. A little caveat, this is not the way you would normally do this. Normally you would have cross-validation. There'd be all sorts of other things to do it. I'm just showing you a simple model here to illustrate supervised learning. All right. So we split the training set or the data into 70% for training, 30% for the tests, okay? And what we're going to do is we're basically going to do a KNN, and we're going to train our model on this 70% of the data, and then we're going to look at this 30% of data unseen and try to predict if they're tumor or normal, okay? Don't worry about too much about the implementation here, because it's just the theory here, and we're checking our accuracy here. And what do we get? 83% accuracy, and it's pretty much got them all. The only thing I really missed is it classified these two as normal instead of tumor, which is not good because these people are sick. But I did a pretty good job. And, the reason I did a pretty good job is it's pretty simple. There's a strong signal in this data, and it's very different in normal and tumors. I was able to pick that out pretty well and do a good job. It wouldn't do near as good a job in a more complicated data set. But this is just to show you how, if you have the labels, you can train a model. You can basically say, hey, these are the answers for this data. If I give you similar data, what should the answer be? And that's all you're really doing. And we'll talk more about that in a couple of weeks when we deal with more of our more classification machine learning models. So let's go on to our main issue of the day, which is unsupervised learning, which is really discovering without these labels, right? Now let's pretend that we don't know which samples are normal and tumor. Can we still find that normal cluster? Can we pull that normal population out of that data set? Well, let's see. So we have our expression matrix, centered and scaled, right? So let's do like last week and do our PCA. We use the PR comp, our expression matrix, and it's already centered and scaled, so these are both false, and we're going to do it on the first 50 and just make a a little data frame to visualize, and we're going to pull out our actual embeddings here. PCA result, remember, to the $X gives us our actual embedding there, right? And then we're basically going to choose, well, out of these embedding, how many should I actually keep? And we'll talk about how we are going to do that. So first, we basically made an elbow plot, a little more fancy elbow plot, and we're going through the first 50 PCs. And we're basically we're looking at the variance explained. So to do that, we're looking at the the different eigenvalues, things like that, proportion of variance explained, and then we're gonna we're gonna have eigenvalues here and proportion explained, and check the drop off and principal component contributions as we go, and kind of look for that elbow. Remember, where's that elbow? Is it going to be around like 1, 2, 3, 4, 5, 6, 7? So like maybe 7, maybe, or 6. It's hard to say, but you could also say this one. And you can take the elbow different places. Your first max, your first change, your last one, your perpendicular. So there's just lots of different ways you can try to figure that out, and a lot of different methods to get this, right? So first we're just building this graph to see it, to kind of conceptually see it. A lot of people just do this visually. So if you can also run the fine elbow point algorithm, and I can do it on the variance and the raw eigenvalues using PCA tools or just regular fine elbow point. And we can also do this rigorous, remember last week, this horns analysis, which is more as a statistical way of finding this. You can throw the matrix there, it'll give you a result there. And if you do all three of those, and run them, here's what we get. Look at this table. It's interesting. So looking at the elbow for percent variance, we got 6 optimal PCs. For the raw eigenvalues, we got 5, and for horns parallel, we got 5. So I feel pretty good. Horns the most rigorous, and 2 out of the 3 are 5. So I say, this example of let's go with 5. I feel good about it. Let's see where that would have been. 1, 2, 3, 4, 5. So that would have been that kind of horizontal one. It's just turn the corner. It's inflecting, you know what I mean? So I could see that, but I mean, it's debatable what it could be, right? So now we're going to extract that number of PCs. I just pulled in the optimal PCS for horn, and from 1 to horn, so that's 1 to 5 PCs. So, and one rule of thumb is when you're dealing with bulk data, you normally have PCs from like 5 to 15, and when dealing with cell data, it's more things like 20 to 50. And we're dealing with huge multi-element data sets, and we're like 50 to 100. So having 5 in our 5 to 15 range for bulk makes sense to me. So now I'm going to plot without labels and then with the labels, right? And here's where we go. It's nice, right? This is what the PCA has given us. And we have some nice variance, especially along PC2, some along PC1, and but we don't really know where anything is. If we didn't have this plot, we would have nothing. Where would you find the normal out of this? Would it be this? I don't know. This? There's no way of knowing, right? It's because the actual truth here is this normal is over here by itself in the corner. So that's kind of sneaky over there, right? So the idea is, can we use unsupervised clustering to find that cluster? Is it possible? I think we can do it. Let's give it a try. So the 2 methods we're going to talk about today are k-means and hierarchical clustering. So starting with k-means first, this is really finding cancer subtypes. And this is really useful when you already know the number of clusters that you should have, which based on biological information, cell types, different things, right? So just to kind of talk about how this works simply, it's worth understanding. So k-means is basically like organizing a party into conversation groups. This is a great analogy, I think. Let's say you're at a party, right? Having a couple of drinks, hanging out with your friends. What you're going to notice is different cliques of people tend to form together, right? You look at a party, you see a cross, you see a guy, it's the quarterback of the team in high school. He's going to have his cheerleaders around him, whatever, right? You're going to have different people that are big cliques. You have the goth kids in the corner. So what's happening is, first, you want to choose the number of groups you want. So I see these groups around the room, and I'm like, I see about 4 or 5 groups of people. Let's pick 5. I want to pick 5 groups or clusters of people. We're going to do this. So what you do is you go to each one of those groups and you pick an initial group leader. He's the guy that's the most talkative, the most influential in that group, and we just guess him in general. And then this is a random pick. And we move everyone in that, everyone joins the closest group, right? So the cliques are going to buy it. There's going to be outliers, but the cliques are going to join the groups, right? And then once they're all in their new group, we're actually going to move that leader back to the center of the group. And then we're going to keep doing that until the center stops moving around and they're stuck, right? And that will be our belief that actually is a real cluster. So just kind of the math behind it is the same thing. We want to partition data into K clusters by minimizing the distance between points and their cluster centers, which is called a centroid. So we want to minimize the distance from the cluster center, pull them in, right? So the algorithm, it randomly selects K points just like we did. It assumes each data point to the nearest centroid, or it's assigning it to the nearest center, and then we're updating to the new people, right? And the repeating steps, it stops moving. And all of this is to, if you see here, this is just the summation of differences across. So you have the summation of how far are you from the center for one cell, sum up for each cluster, right? So we just add them all up, and then it gives us a within cluster sum of squares, where the lower the value, the better, obviously. So really, in plain English, we want points in each cluster to be as close as possible to their respective cluster centers, right? All right, let's run this on the bulk TCGA data now, right? So what do we got here? So first we have our PCA coordinates, right? That was our embedding that we chose, from the Horn analysis, right? So so we could do several things here. We could put our actual PCA coordinates. We could do UMAP, which we'll talk about next week, or we could just offer the expression matrix. If we wanted the expression matrix, we were basically using all the dimensions. But our whole goal is to reduce the dimensions down to get the best signal, right? So that's what we've done for our PCA coordinates, and we'll input in here, and it's just K-means, coordinates, the number of centers, the number of restarts. It just kind of restarts because it moves through kind of a statistical method, and wherever it starts, it can make a difference, right? So if it is at 25 times, it can just kind of average the results. You get a better result. Within reason, it just takes longer. In centers, that's the number of clusters. We're going to guess 3 just to get started. And when we run this, what we can do is you have your KM3 is my result, right? And $ cluster gives us our cluster results. So we see 3 clusters, pretty much rounded out, 1 small one, right? And the within cluster sum of squares is around 5,000, which is decent. But these are more comparable among different methods when you're trying to optimize. And then we're actually going to visualize that. We just made a PDF. We added the cluster assignments, and then we just basically called the PC values that we know for X and Y, and we colored by cluster and shaped by our tumor type right here. And we basically made these ellipses. These aren't clusters. I'm not finding clusters. This is just saying basically a cloud where 95% of the data should be expected to be in. That's all it's saying, right? So basically, if we see here, look at my clusters here, red cluster, green cluster, blue cluster, and it's pretty good. They've done good separations. And really the tumor data is likely to have multiple populations too. So this isn't necessarily one cluster. It could be different things. I mean, who's to say what a cluster is? It's an artistic thing. There's no right answer. But what we do see is we've been able to grab the normal population with only 1 miss, which is what we really hope to do. So that felt really good about it. So we're really seeing, in that cluster 1, we're almost we're just missing 1 off, right? And then we look at both of these, that grabs all our tumor. So we chose 3, but how do we know actually how many to pick, right? So like always, do the elbow plot, right? So I basically made a function where all I did was run k-means a bunch of times in a row from 2 to 15. That's all I did. And then I got that number, that WSS number, right? And then I just plotted it as an elbow. Simple as that, right? And this possible is just one of the ways that it could be done based on what we showed before and possibilities. So really, this could be anything. It's hard to say 3. It really feels more like 4, 5, 6. It looks more 4, 5, 6 to be than 3, right? But it could be a lot of things. Probably 4 is the best thing. But it's hard to say, right? If you do the optimal elbow thing, it says 5. We could also use the gap statistic. This is a more statistical method. So if you're looking at things like elbow, silhouette, things like that, those are internal metrics. They only rely on what we have, that cluster's metrics. While gap actually provides a statistical component to this, where it basically compares. It'll do your calculation, and then it'll basically randomize the data or find random data, do that clustering as well then, and then see basically compare that null distribution to our real one and see if there's a statistical difference. So not only is it better over random noise? That's what GAP's doing for you, and it's giving you a really good result based on that. It's the same thing. I basically ran a function here a bunch of times. K max is 15, so 2 to 15. I ran the function. I did the Monte Carlo, which is how many times it goes over and over on its iterations, and then I plotted the plot. And what you can do, you see, if you have this fviz gap stat, you'll automatically, it can plot, it has several different parameters that can plot. It'll do the gap result, but it'll plot finding the maximum in different places, because it can be found by gap. It can be found by a lot of different things. This isn't necessarily for a gap. This is by first local maximum divided by standard of error. So there's lots of different ways this actually point can be chosen. This isn't actually the gap number. If I actually choose the gap number here, once again, I can pull many different methods. There's like 5 or so methods to get an answer. I am using the Tibbs max because it comes from the actual gap stat paper. I've used other ones and got 3. If I use this one, I get 4. And that kind of feels kind of good here, that first start off turn here, right? So it's possible. So this is the local maximum over standard of error. This is by the actual gap stat call on Tibbs. All right, so they're saying 4 here for the gap statistic. So how about silhouette analysis? We've done that before. I'm basically just running it again here a bunch of times and finding the silhouette for each one of those. And doing a silhouette plot, same as I had before. And we're looking at the highest number is 3 once again. So we're seeing a lot of 3s. And then finally, as a tiebreaker, we're going to do the Jacquard stability. This is under actual good statistical measure. We'll do a bunch of bootstrapping to try to figure this out and get a good result for it. You basically use this FPC package, cluster boot. I put it in the PCA chords, the Monte Carlo. It's always boot for us. I am saying I want to try 3. C42 runs 50. And basically cluster method, FPC k-means. So it's all k-means, right? So I'm doing a 3 on k-means, a 4 on k-means, and a 5 on k-means. Basically, I get my results, 3, 4, 5, and we look at the Jacquard stability in between these, right? And basically anything over 0.7 is pretty good. Anything over 0.8 is solid. Below 0.7 is not good. So here we have 3 solid ones. We feel good about those. For this one, we have 1 bad one. For this guy, we have kind of 2 bad ones. So 2 bad ones, we're going to throw this guy out. It's mixed, some robust, some spurious. And see, the mean's pretty close, so that's not going to give it to you. You got to look at each cluster. So we throw out that one. And the 4 one, we have one that's too strong and too weak. You know, these are good, these are weak. It really feels like this is the best overall to go with 3. So once again, we're seeing 3, so we're going to choose 3 overall. It's the best one. And then also, can we just effectively go for tumor normal, which is going to be k equals 2. If I use 2, I get a solid split. I'm just missing that 1. So we're still able to split just specifically between tumor normal, which is what we really cared about in the first place, right? So that's all of k-means clustering. Now let's move on to my favorite clustering, hierarchical clustering, building a cancer family tree. So here to really understand hierarchical clustering, unlike k-means, hierarchical clustering builds actually a real tree. It shows relationships among all the different levels, right? So everything's connected in a linear, not a linear, but in a tree-like manner, where close relatives, similar samples merge first, and distance relatives merge last. And you can cut the tree at any height to get how many or clusters you want. And that'll make sense here coming in a second. So really, hierarchical clustering builds a tree of clusters called a dendrogram without requiring you to know. It doesn't need to know the number of clusters firsthand. It finds it itself, right? And uses 2 approaches. This agglomerative, which is bottom up. You basically start with each point. Basically, every point's a cluster. And then you combine the 2 similar ones. Now you have half as many clusters, right? And then we want to once again merge similar ones. And then once again merge similar ones. You can imagine at some point, you know, everything's going to be in one cluster, or completely one cluster. That's the top of the tree. And then how you split it up going down is how the tree would look. That's the way to do it from bottom up. Top down, you start with all in the one cluster and split it recursively. Advice of top down is not what you're going to see very often. What we're going to work with today is the narrative, and that's the one you're going to see used in cancer research the most. So pretty simple. You start with data point in its own cluster, identify the two closest clusters, combine them, and then just keep doing that until they're all one cluster. We just talked about that. So there's two different metrics we use for distance with H-class. One, we need to make a distance matrix. We talked about distance matrices a couple of classes ago. We were basically computes the pairized distances between all the points and locks them in a matrix to preserve that value in the future algorithms. And remember, we had things like Euclidean, Manhattan, distances, things like that. And that's distances of the points. But how do we know the distances between actual clusters? So we're in the middle, we're like trying to look at the distances between different clusters. How do we know that? Well, there's 4 different methods for that as well. Or not as well, but there's 4 different methods. So there's single linkage. So you would choose the distance between the closest points. Complete, it's the farthest points. Average, the average distance between all points. And you're just summing these, right? And the Ward's method, which minimizes variance when merging, right? And it can basically merge clusters that result in minimum increase in that total within cluster variance. I like Ward 2. It's pretty useful, especially for copy number and mutation data. And it really gives you a more statistical measure of that, but it's not always the best, and every data set's different. So let's look at our tree. Let's look at our dendrogram, right? So let's build our matrix, the PCA coordinates. We have to use Euclidean because we're using PCA. Dist forms our distance matrix, right? Just like last week. Then we'll put this into our H-clust. H-clust is our hierarchical clustering algorithm, our package, our function. We put our distance matrix, and we choose our method of distance. I choose Ward D2. That gives us our object, and then we basically plot that object. And bam, a tree. Cool, right? That's how you make a tree. And we see a bunch of clusters. Jumping off, I see 1, 2, 3, 4, really 5 clusters I'd like to see, right? But you can also see 1, 2, 3, 4, and you can also see 1, 2, 3, right? Any of them can be right depending on what you're trying to do. So I also see a tumor here hanging out with the normals. So something's going on with this tumor cell that's pulling it across. It might be a transitional cell between normal and tumor, who knows? But it's a very interesting cell, or sample. So now how do we choose the clusters, right? We can do it different ways. We can choose by the number of clusters. If you choose like k equals 3, I'll choose the first 3 splits. 1, 2, 3, right? And if 4, 1, 2, 3, and it'll be kind of hard, probably 4 because it's higher. Or we could choose to cut the tree, right? Basically a line. So you can look at the distance. Let's say we choose 60, and we basically cut the tree at 60. We draw a line, we cut the tree, and everyone left at the bottom. We cut this all falls. That's 1. We cut this all falls. That's 1. Cut this, it all falls at 1. So you would get 3 different trees if you cut at 60. That's how you cut trees. Let's take a little bit nicer dendrogram to look at. We can put the object in as.dendrogram to get a dendrogram object. We can lateralize that object, which just kind of angles the tree to where it's like it's not jagged, it's all straight up left or right. And then we can basically color these. We can add different colors for each of the branches, and we can also label the actual samples themselves. So now you see I've cut out. You can see 3 nice clusters here, and the samples are labeled. These are all cancer. These are all normal with that 1 tumor one there. So cutting the tree. How do we cut the tree? Well, we use the cut tree algorithm, right? Cut tree, HC, the object, and cut it, k equals 3. K equals 4 for 4 clusters. That H equals 60. If you put an H, that gives you the height cut, right? K gives you the number of clusters. So if we use the 3, we will see that here's our split. And once again, we had 11 in that one misplaced one in there. So 10 normal, 1 tumor. And the other 2 are exactly the same as K means found. It's a pretty simple example, so we kind of expect that. But how do we choose the K if we really wanted to choose the K? Let's try a different couple methods. We could use the elbow plot, we could use gap statistic, we could use cluster boot. Just want to show you some different things. Let's try Silhouette and Kalinske, Habermas, and try those out. So I basically am just making a table of all those values. I'm basically running it for values 2 through 15 once again. And so 2 through k max, 2 through 15, and I'm basically making a matrix here, and I'm basically running it. I cut tree on each one of those first. So I take each value, cut the tree, take the CL value, put it in, calculate silhouette for each one of those, calculate Carnot for each one of them, throw them back in a matrix. And we see here, we look for our highest values, highest the better, and we see here the best silhouette is found on, the best silhouette is here, 3. Best CH value is 4. So 3 to 4. 3 to 4. Same things we kind of been saying. So silhouette 3, complementary 4. And you can kind of see here that silhouette tends to favor fewer. CH is more, which kind of makes sense when we have 3 to 4 more. CH is okay with bolt, not too great with single cell, as we'll see next week. But the important thing is to try as many metrics as possible to be able to get your belief. And we'll try the Jacquard stability one more time. Same exact thing, except we put H cluster here instead of K means. And we get our Jacquard clusters. And, K goes 3, it looks a little rocky, 0.57, not loving that. This one is pretty okay, but two kind of low ones. And this 55, 69, 5's thrown out, 3's thrown out. I think you have to choose 4 here. C is our default choice we've been using, and 4 might be a little bit better and might give us a little more granular subtypes. So once again, we're going to cut the tree with 3 here, and take a look at that. So I basically just cut the tree. That was our one that we expected, and we were able to get 3 values here. I meant to do 4 here, sorry. I meant to call the 4th one here. But let's just check out what that would look at. Visualizing across results. Because I want to see show you what that would look like here. This is good. I'll show you how to change this anyway, right? So I want to just change that to 4. So K equals 4 this time, and then and rerun it like I meant to in the first place. All right, here we have our 4 results, right? 1, 2, 3, 4. So now, and these are splits that we kind of expect to see, right? These aren't crazy splits to see. So a little more granular subtype across the tumor cells, right? So you can see how that kind of makes sense, right? So there we go. So we visualized our other H. And let's actually make our expression matrix here. So what we want to do here is we want to keep our clusters we found using the H cluster. Or anything, this will be the H cluster, but it could be any type of clustering, whether it be the H cluster or the K means, whatever. We basically want to keep those clusters as our columns, and then we want to look at our gene expression and do clustering, do hierarchical clustering on the gene expression with our set columns to find groups of genes that are relatively, differentially expressed, not differentially expressed, but just especially strongly expressed in certain clusters, right? So first thing we're going to do is take that gene variance and get our top 50 genes. Remember we had 500 before here. Now we just want the top 50. We're going to take our expression matrix and subset once again for the top 50. So all we've done is we have our same expression matrix, but only the top 50 genes. And basically we want to keep the order of our H cluster. So I've looked at the order. This is the order of the clusters, actually, the exact order the clusters are in. And I basically made the heat map that same order of clusters. And then I've just made basically a vector of the clusters in order and a vector of the colors in order. So like tumor, it's going to be like, the tumor normal, and the clusters. And then just so that'll line up in order, okay? The annotation is going to be kind of complicated, but just trust me here. You can take a look at it. I'm making a column sample, like a sample vector and a color vector, and they match. That's all I'm doing. And then I'm actually making a gap between the clusters, and then I'm actually going to put that expression in, and I'm going to use, now that we have our expression, I'm going to use word D2 for our distance, and I'm going to cluster the rows by correlation. I'm not clustering the columns. So our columns are the exact H columns. So the samples are in order, just like they are in the H diagram. And then we've just basically labeled the normal and tumor like we do with that. Remember that dendrogram we labeled? Just imagine the top is just that colored dendrogram. Cluster, 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 label samples. That's all it is. That's all a heat map is a dendrogram, a tree, right? So we basically just brought our tree and set it over here on the top. And then on the left, we have all our genes, right? And we were allowed these to cluster in an unsupervised hierarchical clustering manner. And look what we found here. It's pretty cool. We found all this great subset here for our normal genes, and this great subset here for our blue cluster, and this great subset here for our red. Now, you could say that red might have another split here, but blue and green are just spot on here. We can look at closer at some of these genes and understand the differences in these clusters. But I think this is a really great result that makes us feel good about our other computational results. And then in general, can an H plus effectively separate tumor and normal? We can put it k equals 2, cut the tree, and here we go. Same deal, just one wrong on tumor. Very similar to the other one. So now we want to compare and evaluate the clusters. Let's look at compare the internal validation metrics between k-mean and H plus, see which one did a better job. So we're just doing silhouette. We basically have our k-means clusters, our distance matrix silhouette, just like we've done in the past. That gives us a silhouette score per cluster and an average. Do the same thing with the HC clusters. Same thing, same average. They're exactly the same because the clusters are the same when it's a simple example. So, and we're also using the same, embedding for both of them too. So it's not crazy that they're all the same. And let's visualize these plots. If using this FC-Viz silhouette, there's a nice silhouette plots. And we can see they're kind of flipped, but they're exactly the same. But this is really good for you to check on with your more complicated examples later. And then I just looked at the ARI, which is the adjusted RAND index. And this is a label invariant belief. It's just use similarity between cell types. At 0.6, really not bad. And they're both the same. So they did a pretty good job, actually. So I feel pretty good about both results, even though it was a simple result. But now you can kind of see how it works, right? Just a foundation of how these two methods work, and you can work on applying it in the more complicated data sets later. There's a few practice exercises I wanted to go through with you. Well, first, we want to compare different linkage methods. So imagine we're doing H-clus like before, but and don't worry about this, I'm just mapping the true and normal labels, basically. I just want to compare these three different linkage methods and see what happens. So I have my PCA coords, Euclidean distance matrix, and I'm just going to run H-clus. I'm going to cut it k equals 3, and then I'm just going to use these 4 different values, and then we're going to look at both accuracy and the adjusted RAND index here. And surprisingly, if you look at the accuracy, complete came on top. Ward 2 didn't do that great. Complete's on top here again. Ward 2 is not that great either. So really, I might have made a mistake using Ward 2. It might be interesting to go back through and use complete as we're getting a better result. So that was pretty cool to look at. So how about the distance matrix? So a distance matrix this is different linkage from the different cluster distances. How about the actual distance matrix from the points? You know what I mean? In some cases, you might need alternative metric than Euclidean. And we know if you use PCA coordinates, we can use Euclidean. We have to, right? Well, let's say we know a different distance matrix might do a better job. Well, remember, like last time we used the CMD scale to try the different ones for PCA. We're going to do this similar thing here for H-clus, where we're going to use the expression matrix and then just use different methods here. And we're going to use Euclidean, Manhattan, cosine, Canberra, just try some different things here. We're also using correlation here as well. And you guys know how to calculate these guys. This is how you would calculate with this proxy function, the cosine version. And then we have all of our, our distance matrices. Just put them in the H-cluses. And we have our clusters basically. And then we basically just cut the tree, K2 for all of those, just for fun, because we're doing like tumor versus normal. And then we're going to compare these different values. Like how well could you separate those two clusters, right? And so we'll look at our CH index first. 1, 2, 3, 4, 5. And we see that some don't work with the real Canberra. I see the highest ones are cosine and correlation, which are well known to do well with gene expression data, especially if you don't use the PCA. And then we'll do a similar thing with our Don index, very similar. We'll try our new things. And once again, if I put in PCA, it kills everybody because that's obviously an advantage to have a more focused PCs. And here, once again, correlation, cosine are winning. Cosine's number one on both of them, I believe, right? Yeah, or at least tied for number one. So I'm kind of look at the H classes on this. Let's look at the dendrograms here. So we see that everything in Pingera except Canberra is really weird. It doesn't do a very good job. But the others are kind of similar. When I see the cosine correlation, I see a little more granular value here. Check these out here. Are these guys here? I'm seeing a few little things here I'm not seeing in these very clean ones that we're seeing. These are almost too clean. We're seeing a little more information. Maybe that's why they're working a little bit better, again, making us a better result. And finally, well, let's say now we have our clustering results. How do we look and see it? How can we validate this? And in real life, you would do a differential gene expression among all the clusters, look at the expression data, and see which clusters, see what genes are more highly expressed or differentially expressed in certain clusters. And highly expressed and differentially expressed are not the same thing. Sometimes you can look at just the higher expression and you're not seeing it. It's not really helping you find your clusters, but the differential expression will do better. But you would hope to have both. But anyway, we're going to manually do this just for fun. We're basically saying cluster 1 samples are everything that's in the cluster 1, and other samples are everything not in cluster 1. And then we're just going to find basically the row means of cluster 1 and means and kind of do a kind of fold change. So we're basically dividing the mean gene expression divided by the other gene expression. So if this number is higher, you're going to get a higher fold change if cluster is over everything else, right? That's all you're trying to do, is this cluster have expression higher than everything else. But this is not differential expression injection. This is a very manual, just very hands-on technique to get a very rough value here. And then we're just basically going to sort these genes. We're basically getting the top 30, top 30 genes basically in each, most unregulated in each one. So basically, we're sorting by fold change, taking the top 30, and these are our top 30 genes of fold change in cluster 1. And then just for fun, I gave you a little way to switch this back to gene signal. These are ensemble numbers. We got them all but a couple. We have the first 29. We don't have 30 genes that are associated with. And I tried to run these through some hallmark analysis, but unfortunately they didn't really, they weren't significant enough to show you. So I basically just kind of give you the idea is later on, the next step would be to look these genes up, see if they're cancer related, do pathway enrichment analysis, and do survival analysis. But we can see, we've got about 5 genes here in this cluster 1, which is the cancer cluster, and they do seem to be very well-known breast cancer-related genes. So there's no p-values here, but we kind of get an understanding of how we could do some validation and genes based on their expression. Some key takeaways from this week, just to summarize, we learned that we're seeing supervised, unsupervised learning. Supervised needs labels. Supervised finds patterns without labels, right? K-means clustering it's very fast and simple. It's good for a quick look over, but you need to specify the number of K, so that's kind of a drawback, and it assumes more spherical clusters, so if they're more elongated, they're not going to be found as well. We have hierarchical clustering. This shows the relationships at all scales. You can cut at any level. It's great for visualization with the dendrograms and heat maps, and it doesn't have to have spherical clusters. And you don't need to specify K. So that's why I think H cluster is a very, very, very powerful method to do a lot of cool stuff with, especially in combination with other methods. And we learned evaluation metrics, internal ones like elbow, silhouette, gap statistic, external ones like cluster boot, and or actually gap stats external, more statistical based ones, right? And we saw how we can validate biologically, which would be always the final result. As I complained about in the past, there's no gold standard for any of this, and it's a very, 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 it's art, not science, clustering. It's 100% art, I guarantee. And there's no right way to do it, and you really need to validate it in biology afterwards to really have belief that it is what it's saying it is. And I had some little quick reference guides, just on K-means clustering, hierarchical clustering, cluster validation, some interpretations for some of the metrics, and that's about it. So for next week, it's going to be a really cool week. We're going to explore more advanced clustering methods and advanced nonlinear dimensionality reduction. So we'll be doing reductions with things like TS&E, UMAP, and then we'll use those as input for graph-based methods like Leighton, Leighton, Gaussian mixture models, and things like density-based clustering from HDB scan, things that you're going to see people using at the cutting edge of genomics research. And then I made 3 pretty useful, I think, practical problems. I worked really hard on these, so I really would think you guys should check them out. I did k-means with a lung cancer data set. I did hierarchical with a colorectal cancer. I did a supervised K&N and showed you how to pre-process single cell data in problem 3. And I did kind of repeat of today's H-class hierarchical, but I did it with single cell data, which I think is pretty cool since we did bulk actually today. And that's pretty much the week 2 session complete, but I just wanted to kind of just briefly show you what I'm talking about with these homework assignments. So for the first one, we're able to download another sample and prepare this TCGA LUAD data for lung cancer and be able to normalize it in every way, get our scree plot, and found our clusters. And I wanted to show you this FVIS silhouette FVIS cluster. Gives some really cool ways of plotting things. You saw the last time I used this for cluster, but you can also do this cool thing. It does a lot of things for you here and gives you a cool, visualization of different clusters and kind of their data clouds, right? And then I did COAD for the second one. I downloaded another TCGA data set, if I could do this right. The COAD one there, kind of very similar. We found the best PCA values, and then we kind of ran it. And with this one, we did other things that we wanted to compare different linkage methods. We checked those out, and then we were able to create a clustered heat map based on some of these results. It didn't work out so well, but you do see some differences, especially among the blue cluster here and the red. There's a little bit differences here, but these are 3 clusters that we were able to just quickly jump through. And then on the I was able to bring from Experiment Hub, I brought in this head and neck cancer data set, single cell data, and I basically built a K&N model for that to kind of show you what's going on with that. And then I also figured out the number of PCA embeddings. So not only were we able to download the data, we were able to pre-process it, get the number of PCA embeddings, and run a K&N classification model for fun there. And you see that, we see a vastly different differences. The elbow wanted 2 PCs, raw elbow wanted 51, and horns wanted 25. These are very different numbers, right? So I think we go with the horns parallel. So we're going to use our PCAs, and then we'll go ahead and plot the results of that K&N thing, and you can see 93% accuracy. Well, that's because I used the UMAP. I used the PCA coordinates for the input as that, right? And the PCA already done all the work, so it basically just was able to jump on that and get a good result on these, especially since the way I did it, there's data leakage here and it's not perfect. But just to show you how to do them, we'll be building a couple of weeks better candidate models. But once again, you can do that if you want. But obviously, if they were 98%, then that's all we'd use. So that's why you can't really trust this result, right? It's more they're just so distinct, and we're doing it on top of PCA. Anyway, so the fourth one is this single cell data set, which I'm really proud of. I worked really hard on. And we're using PCA, which is kind of interesting because I don't normally do that with single cell, but here's our PCA. And there's all of our different, I think 9 different cell types here. There's 3 main ones, the cancer cells, the T cells, and the fibroblasts. They are the majority of the cells. And we're going to try to do our clustering and see what we can find. So what we're really going to do is try to find the broad here. We just want to look for these 3 populations right here, just to make it easy for you. So we're doing k-means on the PCA cohorts, 3 centers, and we visualize that, and lo and behold, we got our 3s. Like, look, so cluster 1 is here, cluster 1's here, cluster 2's here with the triangles. This is cluster 3, right? So, and if you look at the colors, T cell, cancer, and fibroblasts. We've nailed our major cell types pretty well. Obviously, the other ones are a lot harder to find. We have to do a little more. We have to really look into this and really mess with the parameters to try to find those. we found our best K, best silhouette, and so on and forth. And we did the same thing on H-clust. And I just wanted to separate that out. I ladderized it, and you see our three things. And I think this is pretty cool. I just did cancer, fibroblasts, and T cells because they're the most. And you can see these split out really well with H-clust. All of our cancers, all of our fibroblasts, all of our T cell. Perfectly freaking split. I'm really proud of these. A little bit of leakage in here, but really good. And our black is just everything else. So there's going to be some black cells coming out of fibroblasts. It's black. It's the smaller cell types that come out of those. And those are we expected to see in these two cell types and not in cancer. So, I think H-clust did a pretty darn good job here. And visualizing the results, once again, we see, T cells, it's a cancer, and we see fibroblasts well separated out. And then if we make it actually embedding, we have our three clusters, and you can see cancer is just solid here, and then we see some small cell types found in the other clusters. And then also what I was wanting to show you, obviously T cells seem to have the most expression here, which is really good because that would be a really good analysis forthcoming. But what I really wanted to show you is for single cell, remember last week I made the cool plot out of the bulk data. For single cell, you do not make plots like this. Look how sparse this data is. You can't see anything. It's useless, right? That's why people find their clusters and then they find like their top 10 most upregulated genes, different expressed genes in that cluster, and they just make a tiny cluster of just those genes in that tiny cluster because they can do some different kind of transformations to get more signal. But that's why you can't do sparse matrix visualizations, unfortunately, right? And then we do some things like we look at the internal metrics. But yeah, guys, check this out if you can. I think you will learn something extra, seeing just some more repetitions of this, and also seeing the single cell data. And we will actually be using that same single cell data the next week, so that'll be pretty cool, I think as well. And so I guess that's it for this week. Thank you guys for listening in. This is one of my favorite topics, so I'm really excited about it. I think K-means is a good, just quick visualization when you think you might know what you're looking at. H-clust is a more refined topic. I think it really organizes the different clusters very well to be able to pick out a lot of genes, especially smaller genes, and it can be done even after different clusters. Like we saw the clustering on the heat map, then we did H-cluster on just the genes. You can do things like that. So I think H-cluster is extremely powerful, and obviously it makes trees. I'm a big phylogenetics person, so it helps make trees too as well, and beautiful heat maps. So hope you guys learned a lot this week, and get ready for next week because it's going to be a heck of a lesson with a lot of information. And I guess with that, I will leave you guys. You have a nice night.